So I came to Libby initially in 2001 because the state medical officer came down where I was studying at the University of Montana and said, we think there's more autoimmune disease in Libby than there should be, and asked for somebody to come up. And so I was very enthusiastic to come up and start meeting the people in Libby and started screening for autoimmune disease at that point. And we have successfully not only shown that, yes, there is more autoimmune disease in Libby than there should be, but also over the last 24 years, we have learned so much about the disease and the disease process to where we believe that the lamellar pleural thickening that Dr. Zanuck talked about is an autoimmune disease. And so we want to talk about that a little bit this afternoon.
The other thing we wanted to do is also explain kind of what autoimmune disease is, and some of what we know, not just from Libby, but from autoimmune disease in the world, is partially why do some people get it and some people don't. We know that autoimmune disease prevalence around the world is increasing, so if it seems to be, hey, I never knew people when I was growing up who had autoimmune disease, and now everybody seems to have an autoimmune disease, it's not in your head. It is that the prevalence is increasing, partially because diagnoses are getting better. We're actually recognizing that these autoimmune diseases exist, and it's not just people making things up or thinking that they have diseases that they don't. They're real. But also, we have lots of underlying causes that have been identified over the years. So we know there's a genetic predisposition. So autoimmune diseases do run in families, but that's not the only thing. So we have environmental factors, we have lifestyle factors, diet, infections can sometimes trigger autoimmune diseases, especially if you have these other predispositions. So here's just some common autoimmune diseases in general. But one thing we've wanted to know for many years is asbestos exposure one of these environmental triggers, one of these environmental causes?
This was a paper that was published in 2018, working with a rheumatologist from Kalispell, Dr. Deagle, and he helped us pull together a whole bunch of cases, but he also helped us really start to define this disease that we see in Libby in terms of an autoimmune. And as I think I mentioned before, we thought there was just going to be one simple syndrome, and we're going to call it Libby autoimmune disease, and yeah, that didn't work out. Instead, it's a whole array of different diseases. And so this paper really goes through that and describes what this disease looks like, and it's different in every single person. Some people have multiple disease presentations. So this is a table from that paper, and what I want to point out are 2 points here. You'll notice the red box. The red box is around the diseases that we call systemic autoimmune diseases, meaning that it basically affects the whole body. The immune system throughout the body is not working correctly. Some of the other diseases like psoriatic arthritis, multiple sclerosis, type 1 diabetes, those kinds of disease we consider tissue specific. And what we have found so far is that Libby, as an environmental trigger for autoimmune disease, triggers the systemic diseases. The other point from this is that it's those diseases, if you look at the far right column, it's those diseases that are elevated in frequency in Libby compared to the rest of the country. And if you look at those numbers, the ratio of the frequency in Libby compared to the expected frequency throughout the United States, systemic lupus is 12 times more prevalent in Libby than elsewhere. RA 6 times, systemic sclerosis 7 times, sarcoidosis 14 times more frequent. So we had a question, how much are we seeing? We're seeing a lot. This is a paper that was just published last year by myself, my colleague Fred Miller from the National Institutes of Health, and by a geologist. She's actually calls herself a medical geologist, and she's wonderful. She lives in Nevada. She's at UNLV. And we put together this paper to answer several questions. And one of those is, what is the public health risk for autoimmune disease from asbestos? And so we created some maps. And if you look at some of the student posters over there, they've actually used that map that shows that amphibole asbestos, rather than chrysotile, we're talking about amphibole asbestos, like Libby amphibole, the places in the United States where you can get exposed to amphibole asbestos, and practically speaking, every single state in the United States has this stuff. So this is not just a problem for Libby. This is a public health problem for this country. And that's why whenever I go around the country and I talk about what I've learned from Libby and about asbestos and autoimmune disease, I always title my talks Lessons from Libby. You guys are educating the world. And we are deeply grateful to this community for helping us do that.
Okay, and so I think this was part of our screening process from the CARD clinic. So we've been working with CARD for many, many, many years, looking at autoimmune markers, so especially autoantibodies. And so this 2024 report, which is showing that our autoimmune disease percent prevalence in the population is 20.5%. So this is above expected compared to our normal or our unexposed populations. And autoimmune symptoms are even higher. So even if you don't have a diagnosed autoimmune disease, you can still have autoimmune symptoms. And this is quite prevalent within the community. And so overall, we think, so from all of these last papers, we really believe that asbestos exposure is a factor for autoimmune disease development. And that autoimmunity in Libby is actually higher in males and females, is what our data is showing, which is unusual. So in many, many autoimmune diseases, we have a 3-to-1 prevalence with female to male ratios. So that means females are much more likely to get most autoimmune diseases. There are exceptions, but it seems that in Libby, we're seeing a slightly higher in males and females. So this is another just piece of the data saying that these diseases are a little more unusual compared to other autoimmune diseases we're seeing elsewhere. So one of the things that my group at Idaho State University is really focusing on right now is understanding the mechanisms behind autoimmune disease development, and especially with the Libby amphibool. So we want to know what the immune system's doing, what are the cells doing, and how are they doing it. So then we might potentially figure out a target that we can go in and therapeutically target. Okay, so just a little bit of background first of how the normal immune system works. So our immune system, we make these antibodies against foreign agents, foreign proteins. So we call these antigens. So things like bacteria or viruses, right? So we bind to them, and then this triggers the immune system to get rid of that protein. And hopefully the bacteria or virus it's attached to. We also have tolerance. So when we have self-antigens or our own proteins, we should have tolerance against them, and our antibodies should not recognize and bind to our own proteins. So unfortunately, with autoimmune disease in general, our immune system starts recognizing our own proteins as foreign. So antibodies are made against these self-antigens, and that triggers then destruction of tissues wherever those antibodies bind. So let's say if you have type 1 diabetes, we mentioned that earlier, the antibodies will bind and attack your cells in your pancreas and destroy those cells, and then you don't make insulin anymore because those cells are destroyed. So we have been testing for several different markers of autoimmune in Libby. And so our anti-nuclear antigens, or ANAs, are kind of our broad scope of antibody detection. So these are antibodies that bind to nuclear antigens like DNA, so things that should normally be internalized in the cell. They're broadly associated with various autoimmune diseases.
Primarily the systemic autoimmune diseases like lupus, scleroderma, rheumatoid arthritis.
Okay. So ANAs are one thing that we test for when we get serum from patients in Libby. Another thing we test for are mesothelial cell autoantibodies. We call them MESCAs. These are antibodies that bind to antigens on the mesothelial cell surface. These are the cells that line the pleural cavity. So we're very interested in that because of that pleural disease that exists in Libby. And we've seen an association of having these antibodies and also having that pleural thickening, that pleural fibrosis that's been talked about. And then the last one that we've been looking at are anti-PLG or anti-plasminogen antibodies. We think these are kind of a subset of the MESCAs, but they bind to plasminogen. And we've also seen an association with pleural fibrosis. So our autoantibody prevalence in Libby, as of, I think this is from our last year's report as well. So we're seeing a 17.9, so almost 18% positive for ANAs, 16.6% positive for MESCAs, and 6.3% positive for anti-plasminogen. And this is across all of the patients that we have screened. And so these are above expected or above our control populations. And so our ongoing research is looking to see if any of these antibodies can be biomarkers for disease, for later disease development, and also looking at how Amphibold triggers the antibody production. So again, potential triggers for autoantibody production, just our general what we know about autoimmune disease. So microorganisms, infections can trigger autoimmunity, as can xenobiotics, so like smoking, chemicals, toxins, pharmaceuticals. And these tend to be what we call molecular mimicry. So antigens from bacteria sometimes look like antigens that are your own. So the immune system gets confused. Other things, some things like pharmaceuticals can alter antigens and make them look different to the immune system. So we'll talk about that in a minute. Apoptosis of cells, when cells start picking up things, especially like asbestos fibers, they start dying, they start putting out pieces of themselves, these little blebs, and that's when they can release internal antigens like DNA or histones that should normally be hidden inside, away from the immune system. So the immune system should never encounter double-stranded DNA or your internal proteins. And now all of a sudden, these cells are kind of releasing them all over the place. So the immune system starts picking them up and recognizing them. So that's basically what I just said, is if we think the cell blebbing is starting to release these autoantigens, it's confusing our macrophages, which are our cleanup cells, and then they start giving these pieces to the immune system saying, hey, these are bad, you should start getting rid of them, and so we produce ANAs against them. So this is mainly how we think ANA production is triggered. What we think is happening is when the cells pick up asbestos fibers, since they can't break them down, they can't destroy them, they actually start causing apoptosis in the cell. So under normal conditions, our macrophages pick up those blebs, they clean them up, it's called efferocytosis, they recycle the pieces, and nothing really happens. But what we think is when asbestos fibers kind of trigger an alteration in the macrophages start picking up and then presenting these little pieces of the cell to the immune system saying, hey, there's a lot of inflammation, there's a lot of cell death, there's a lot of bad things going on, here's all the stuff that we picked up at the site of that. But what they're really presenting are your own pieces of your own cell. So they're confusing the immune system and causing them to make these ANAs. So happy to answer questions later if that didn't make sense. And so what we see in our cell culture models when we treated macrophages with Libby amphibole versus our untreated cells is we saw a decrease in this efferocytic capacity. So this quiet cleanup, this ability to take those blebs from those dying cells and get rid of them and not cause an immune system or an immune response. So what we're seeing is a decrease in that ability. So we can see this in here. This is how we count. So I have a student who literally sat there and counted on the fluorescent microscope how many dots, how many green dots they see in the cells. So we see Libby amphibole skewing these macrophage functions. Okay. So cell apoptosis, or this dying of the cells, can also result in inflammation. And so we think that this inflammation can lead to changes in our immune responses and our self-antigens. So again, reminder, our tolerance to our own antigens, so we should not recognize those. But if these antigens become modified, so when we say that, we're thinking that chemical groups get added or changed in them, so the molecular structure looks different, and now these antibodies start binding and recognizing those self-antigens. So for our mesothelial cells, we can see here are normal cell surface proteins. Our immune system usually doesn't recognize them, ignores them, says those are normal self things. But when we get stressed, when you have lots of immune system responses, when you got lots of inflammation, these proteins can get modified or changed, and then our immune system starts recognizing them and making antibodies against them. So we think this is where our mesothelial cell antibodies and our anti-plasminogen antibodies come from.
All right, thank you, Kinta, for that. I hope that made sense to everything. I think that was a really good explanation. So one of the questions that everybody always asks is, we're seeing 20% maybe of the population with autoimmune disease. Why isn't 100% of the people exposed? And of course, as Kinta pointed out, there is a genetic component. There are other things that can trigger autoimmune disease, et cetera. And so we don't know the answer to that question. But we would like to know what it is maybe that's protective. Maybe we can use that protective idea to help people. So an individual's DNA, meaning their genetics, and their own immune status. For example, have they recently had a viral infection? Have they recently had a major surgery or a major trauma? Those things can also contribute to your risk of an autoimmune disease. The extent of the inflammation can also affect antibody production. And so the inflammation signals helps wake up and worry the cells that make the antibodies. So they make more antibodies. And so a lot of inflammation can act like what we call an adjuvant, something that just shakes up the immune system and gets it going. And so some of those can be, as we age, there's more inflammation. Nutrition can affect our inflammatory status. Different infections that we've had can affect our inflammatory status. And so there are a lot of other things that can raise your risk. And probably lots of other things we don't even know about yet. So that's one of the things that I always talk about researchers as being okay with not knowing. A lot of people who are not scientists struggle with that idea of not knowing. I want the answers now. Well, we want them too, but we know that we don't have them all. We try to be patient. So that's it.
Have you seen this disease, more in different blood types than, I'm O positive. Have you seen anything in O positive, B positive, A, B, C, or D?
As far as I know, there's been a lot of studies looking at different infection rates and different blood types, different autoimmune responses, and I think the overall consensus is blood type doesn't really affect too much. There might be a slight increase here and there for different things, but it doesn't seem to be enough of a factor for us to really say that, avoid this if you're type A or do that if you're type O.
Yeah, Dr. Servy's been involved in, and I have also been involved in a couple of studies looking at how can we prevent the disease. So we did this all in mice, and the mice were given a flaxseed extract. You all have probably heard that, flaxseed, so good for you. Well, these mice were given quite a bit of flaxseed extract, and they actually did really well. So antioxidants are always good for you.
CARD gets a lot of questions from patients asking who gets autoimmune disease, who gets pleural disease, and are there people that get both? Do you have any idea what the prevalence is it either or? Is it usually both if you get one or the other?
Thank you for that question. That is a question that we have been asking for a long time. It was one of our original research questions when we first got started, I think in 2009. And we hypothesized that we would see more of the pleural disease in people that had the autoantibodies. We believe that it is an autoimmune response. Unfortunately, the data is not supporting that. So let me explain why we think that is the case. I truly believe that the autoimmune condition does affect the disease, especially the lamellar pleural thickening. We also have seen ANA frequencies being pretty high in the people who get that disease. But the actual scientific correlation has only really popped up and been evident in people with the very progressive form of the disease. So we talked earlier today that some people can live for quite a long time with the disease and not have it progress a lot, but others have this more rapid progression version. And when we look at those people and their autoantibodies, especially the mesothelial cell autoantibodies and the anti-plasminogen antibodies, and also the ANAs are all higher in the people who are struggling with the more progressive disease. So we think that the reason we don't see a good correlation between the presence of the antibodies and just simply diagnosis of lamellar pleural thickening is because it is a subset, a rapid progressive subset of those people with lamellar pleural thickening, where that it's actually an autoimmune progressive form of the disease.
Yeah, and so another project my lab's looking at right now is trying to look at the mechanisms of those. So when you have these mesothelial antibodies or these plasminogen antibodies, once they bind to the cell, what are they then doing? And so we have seen some correlation in our cells with increased collagen, which is a part of the fibrosis process. So we think there is some form there, but it could be you also just have a lot of inflammation and autoimmunity together, and maybe that helps drive the disease. So it's not necessarily one causes the other, but it might be several things working together.
I know a gentleman, and his brother ended up dying from the asbestos. He doesn't have it. Have you gone in and looked at siblings, mothers and daughters, mothers and sons? You know, if one has it, why didn't the other one get it? In a family, you know, what's that one person not have that these other three or these other two did?
Are you asking about the autoimmune diseases or the lung disease?
The lung disease. Oh, and autoimmune too.
Okay.
I mean,
So when we do our screening, we are blind to who we are screening. So I will get a number, a 6-digit number, an age, and I think we get sex, so female, male. And so my students get that, and they get a blood sample, and they screen it. And that's all we really have to begin with. Later, we can go back and add some more data points in there, but we don't actually go back and look and say, this is from Susan, and this is from her daughter, Meg. Like, I'm actually not authorized to have that data. I'm not a physician. And so patient privacy, we don't want that data. And also then we can analyze it without any bias of like, oh, we think this family should have some, so we should look more carefully at this group and see if we can find something. So we're just looking more broadly, at least from the research side.
So there's a group in Cappadocia, Turkey, a similar type of patient population as Libby, where they're exposed to this, I think it's called erionite, which is a type of asbestos. And Miguel Carbone is the researcher out there where he looked at it, and there's some families who were very genetically similar who get the mesothelioma or the cancer from it, and then other families, even if they're related, but somehow they determine they're genetically not the same, that didn't get the cancer, even though they're all exposed to the same thing. So there's a genetic predisposition, but the point is that if they were never exposed to it, they would not have gotten cancer. And naturally, the first thing that these big, I want to call them pro-asbestos people are going to do is try and blame the victim. Well, you had this genetic predisposition, but if you didn't have the exposure, you would have never gotten the cancer.
I was talking earlier about how over 90% of CARD patients agree to participate in research, and one of the things that you can agree to, we ask you for about 7 different things, and one of them is agreeing to genetic research. So if you give us a blood draw and you say yes to genetics, we keep that in our negative 80-degree freezers, which will make it viable for a long time. So that sort of research is possible in the future, even though we haven't done it yet.
This is multifactorial. There's genetics, there's environment, there's lifestyle, there's all these other things. So it's hard to do studies when you have too many variables. So up to now, we've kind of looked at each variable separately, and genetics would be another variable. But to do a study where you're looking at all these variables at one time is a really hard thing to do scientifically.
I want to say a comment. You know, all of these questions are very, very important, and you're raising points that are really, really important. The fear is that we don't know if this is going to happen or not. If they close the clinic, this goes away. So this is the this is a really call to the community. I think the community is the roots of many of these things that have happened. And I mean, we all have to put together an effort for the clinic to continue. All of these questions are so real and so painful for all of you, and so painful for all of us because we feel it. And we're afraid that nothing of this is going to happen if the clinic stops. So may this be a call to really, put all our efforts together for support to the clinic and tell the authorities this is real, this is happening. We feel it in our blood, in our families, and we want the clinic to continue. And I think this was the purpose of really bringing you all. I'm not finishing the meeting, but bringing you all together here to really tell you you have support from the science side in New York and Montana and Idaho. We need the support from the community, and we have to work all together for this not to finish. It's very scary, and your points are so true and so real, and we want to make them happen. But we need your help. We need the help of everybody.
And we do appreciate all of you who have consented to be part of the research and given your blood and given your x-rays and whatnot. And so hopefully we can answer some of these questions in the future. I think genetic screening is just getting better and better all the time. So maybe we can answer that soon.